Okay, we're going to talk about another class of genes which are involved in cancer. This time it's the oncogenes. We've talked before about the tumor suppressor genes. Now, the tumor suppressor genes, when they stop working, and those genes become non-functional, that's when tumors tend to arise. The oncogenes are almost the exact opposite of this. Right. Now, an oncogenes, we can say either excessive expression of the normal gene or expression of a mutated form with an oncogene that will lead to tumor formation. So it's when these genes are switched on that it tends to cause tumor formation, and it can be that it's perfectly normal gene product, but there's just much too much of it, or it can be a slightly altered form of the gene product, which isn't quite doing the job it's supposed to be doing, as we'll see in a bit. Right, let's just refresh our memory about what cancer is. So here, what I've drawn is I've drawn some epithelial cells, and maybe lining the colon or lining the ducts, the milk ducts of the breast, places where cancers arise. So you have a single layer of cells, and when one cell dies or it gets removed by some kind of mechanical abrasion or whatever, well, the ones next to it, they will divide and sort of fill the gap. So that's a normal repair process that's got to go on. What happens in cancer is the cell division, the controls of cell division go, so the cells will start dividing out of control, and quite often as they divide, they'll lose their sort of normal sort of flat shape, and they'll become sort of look like a normal cell, like sort of non-specific round shape. At the moment, that tumor isn't what you'd call malignant. It's benign because it's just staying where it's grown too much, but it's staying in the right place in the body, on top of that blue line, which is the basement membrane. However, if it goes one step further and it becomes malignant, then it might start dividing below the level of the basement membrane. It will invade the basement membrane and divide here. And then this thing I'm trying to draw here, this is a capillary. So you've got your endothelial cells of the capillary, with a couple of red blood cells in there. And maybe one of these cancer cells could get into the capillary, get into the bloodstream, and then spread or metastasize in the body. Okay, so let's have a look at oncogenes. And historically, I'll show you where they come from. Now, historically, they come from a cancer in chickens called the sarcoma, which is caused by a virus. Now, I don't want to mislead you. Viruses can cause cancers in humans, but all the common major killers are not caused by viruses in human beings. They're caused by different things. But let's talk about there's a virus here called sarcoma virus, and it affects fibroblasts in chickens, and it makes the cells grow out of control. Now, when they looked at the genes, and that they found there was a gene, then they called it SRC, and they called it VSRC, and it was this gene. This was an oncogene. The expression of this gene, what it did, oncogene, it made the cells divide inappropriately, and it made them round up. What I mean by that, they lose the shape that they're supposed to have, and they just become these sort of general blob-shaped cells, and they just divide like mad. So there was a gene in the virus which was causing that, and I think the idea for the virus was that if it could make more cells divide, then it could infect more cells and it'd be more effective at spreading itself. So it was an evolutionary advantage for the virus. Now, it turned out where this virus had got this gene from, it had got it from the chicken's genome. Now, the version of the gene the chicken had, they call it C for cellular rather than virus, is c-src, and this is a normal protein which is involved, when that's expressed, it's involved in promoting cell division. But it's not on all the time. It's only switched on when cell division is appropriate and it should be occurring. And what the researchers found was the viral version of that gene, the v-src, that was missing a bit at the end of the protein. They're called the C-terminal end rather than the N-terminal end, missing a section from the C-terminal end, which enabled the activity of the protein to be switched on and off. Okay, so in the viral version, the viral version was on all the time, and so it made cells divide all the time. So that was where the first oncogene was discovered. And usually oncogenes are given these little names, C something, so it's a cellular, and then like a little 3-letter word afterwards, a 3-letter acronym. So let's have a look at another one, another oncogene. So that oncogene there is it can cause cancer in normal cells if it gets mutated into an active form where it's active all the time, or if it's overexpressed, it can cause cancer in those cells. Let's look at a different one here this time. This one is called c-myc. Now, c-myc is the gene codes for a transcription factor. And a transcription factor, you'll see in an earlier another video, you'll see that switches on a gene. It makes RNA polymerase bind to it and makes RNA from it. And so if you have, here's a gene, I'll draw it here. This is the 5' end of the gene, this is the 3' end of the gene. You'll have this bit here, which will be the coding. I'll draw a couple of introns. Okay, so it's got introns and extrons, yeah. The reds are the introns and the blue bits are the exons. But upstream of it, the 5' end, you have this region here. It's a specific sequence called a promoter. And transcription factors bind to the promoter and they switch on transcription of the gene. So c-myc is present in cells normally. It's a transcription for lots and lots of genes. I think I read it was like 15% of all the genes in the human genome use c-myc as a transcription factor. So it's a normal gene, it needs to be there. But when it gets overexpressed, so that means you make too much of it. And it can happen that the gene gets duplicated, or the control of the c-myc gene goes awry. You get too much of it, and you get too much transcription, and the things that the gene products that it might be transcribing may cause cell division, and you get out of control cell division. So c-myc is another one which can cause that. One more example. It's another very commonly found one called c-ras. Okay, I'll do a different color. So c-ras, and ras is like Sarc. There is a version of it which is found in a virus, I think. But anyway, c-ras is a normal protein. And it's called a G protein. It binds, what it does is it binds GTP and it hydrolyzes GTP to GDP. Now, when GTP is bound, the ras protein is active. When it hydrolyzes the GDP, it's inactive. Ras is involved in promoting cell division. So when GDP is bound, it's going to promote the cell to divide. Now, in a mutated version of the protein, mutated form, it loses the capacity to hydrolyze GTP to GDP. So that means that the ras protein is always on. It's always active. It causes activation of pathways which promote cell division. Inappropriate cell division, cancer is going to occur. And one last one, there's another oncogene is, this is one, the normal version of the oncogene, it codes for the receptor for epidermal growth factor, which is called EGF. Epidermal growth factor does, amongst other things, one of the things it does, it's like its name suggests, it promotes the growth of epidermal cells, that makes them divide. Okay, so here's the phospholipid bilayer, and here is the receptor for EGF. Receptor. And okay, now the EGF, which I'll draw as this red blob here, this is the hormone. It can't pass through the lipid bilayer, so what it does is it binds to the receptor, and then at the other end of the receptor you have enzyme activity. And that enzyme activity is only active when EGF is bound. And what that does is it causes changes here, and it switches on other genes. It switches on genes involved in proliferation, in cell division. There's a mutated version of the EGF receptor gene, okay? So the mutated version is on all the time. Like the RAS thing, it's always on. So it doesn't matter whether you've got EGF bound or not, it just keeps switching on. So that means that it's going to switch on all these genes all the time that are involved in cell division, inappropriate, out of control cell division, leading to tumors forming. Okay, so we've got oncogenes there, which are almost the complete opposite of tumor suppressor genes. Tumor suppressor genes keep a lid on cell division, so when they get damaged and they stop working, the tumor can grow. These oncogenes are involved usually in promoting cell division at appropriate times, that you know that you need them there. But when their control becomes damaged, either you've got too much of them or they're switched on all the time, then they create this runaway cell division.